Okay. It is my pleasure to introduce Dr. Luke Iwanowicz. He was introduced to fish health while volunteering at a recirculating aquaculture facility while working on his undergraduate degree at the University of Massachusetts Amherst. He then moved to the University of Arkansas Pine Bluff, Old Golden Lion, to study aquaculture with an emphasis on fish disease. It was there where he developed a passion for virus discovery. After earning his master's degree at UAPB, Luke worked for the USGS National Fish Health Research Lab in Leetown, West Virginia. Soon thereafter, he enrolled in a doctoral program back at the University of Massachusetts Amherst, researching the application of fish health metrics as indicators of environmental health. He continued to work at the USGS, the center which became the Eastern Ecological Science Center, as a principal investigator, focusing on the application of resident fishes as sentinels of aquatic ecosystem health, the development of molecular tools for field study, and the identification of emerging diseases of fishes. He also established and managed a core technology team to support programmatic needs relevant to environmental water sampling for biological activity, including endocrine disrupting activity and toxicity. And then just last year, Luke moved across town to the USDA ARS National Center for Cool and Cold Water Aquaculture, where he is now a research molecular biologist. And we'll be presenting today on the next generation sequencing approaches for pathogen discovery. All yours, Luke.
Okay, well, thanks, Nick. Thanks for the introduction. So today I want to present some strategies, kind of give a little background on the application of next generation sequencing, specific for identifying pathogens and kind of describing microbiomes and environments. As Nick mentioned, part of my background is investigating fish health in the environment. So this is a tool and strategy that we adopted, in the last, well, I guess the last decade or so, and have really utilized this tool for asking, some fundamental questions about potential causes of disease and anomalies. Okay, so here's just a simple roadmap of where I'd like to take us today. I kind of want to give a brief history of sequencing technology, introduce some next generation sequencing platforms. NGS, I often joke, I refer to this as now generation sequencing, because essentially this is where we are these days. Some samples, collection considerations, library prep strategies, bioinformatics, and then kind of interpreting data. So this is I thought this might be a nice place to start, right? This is essentially 5 p.m. on Friday before a holiday weekend, most likely. But anyway, fish kills happen. The public sees this, fish managers see this, and people want answers. External lesions on fish, same thing. Things that are very obvious to the public, we need answers. And depending on whether we're looking at fish in the environment, fish in aquaculture settings, essentially we need to employ tools to provide these answers. This is just a really simple slide. There are many different causes of fish kills, right? Not always pathogens. Disease outbreaks, algal blooms, low DO. In any case, arriving at that answer is something we critically need to do. Back when I used to work in Andy Goodwin's lab, it was not uncommon for fish to show up in the diagnostic lab, and we not identify a pathogen or causes for fish kills. Andy used to jokingly scratch down those interpretations as loss of validity, which is a fun term, and very likely that's not the case. Most likely we just didn't have tools to ask the right questions. So classical disease diagnostic approaches, again, it's just fairly simple: histology, parasitology, bacteriology, classical methods that many of us, if not all of us, learned at some point. You know, a lot of these approaches require us to have some idea of what pathogen may actually be present. You know, kind of in this here, kind of transitioning into molecular approaches, but you know, again, with PCR, we need to you know, know something about the sequence of potential pathogens or microbes that we're looking for. I really don't want to get too heavy into the history of sequencing technology, but I do think it's useful to kind of quickly skim through sort of the beginnings, right? So Maxim Gilbert sequencing about almost 50 years ago transitions into Sanger sequencing, with which all of us, I'm sure, are familiar with. You know, and then basically at the turn of the century, you know, there were some considerable improvements in sequencing technology that basically allowed us to start forming de novo sequencing. You're basically sequencing DNA without having a priori knowledge of the sequence present, and the throughput of some of these technologies, you know, really increased to a level that we're really challenged, and still does challenge computing methods. So those of you that are involved with next-gen sequencing are probably familiar with Illumina sequencers. I mean, these are pretty much the industry standard for next-gen sequencing. Due to their high fidelity and high throughput. You know, if you notice, you know, the output in some of the cases here with NovaSeq X, you know, we're looking at, you know, terabytes worth of data. You know, numbers that are just staggering, and obviously need significant computing power to actually to handle. So advantages of second generation sequencing are the high sequencing accuracy. You know, these days is relatively inexpensive. You know, cost used to be a huge barrier to high throughput sequencing. There are a number of disadvantages, including the short sequence reads, and you know, it's really difficult to sequence full genomes if that's even a goal. We now have third generation sequencing options out there. Pacific Bio has a real-time sequencing method. You basically, in these cases, now sequence up to you know, 125,000 bases instead of 100 to 300, like the short reads. And there's another technology out there by Oxford Nanopore, which is sort of the primary competitor to PacBio. These are primarily the two technologies used for long reads and third generation sequencing. So advantages of using these methods are basically long fragments can be read. Now there actually are options for sequencing full-length cDNA. This is sort of a recent advance in this technology. You know, in the case of Oxford Nanopore, these sequencers can actually be brought into the field or into labs off-site. So there's significant high portability if sequencing has to be done on the spot. You know, the disadvantages, as I was working on this slide, interestingly, the fidelity of long reads with both of these technologies, with PacBio HiFi and Oxford Nanopore, with the advance of the R10 chemistry, these methods are basically on par with next-gen, with the Illumina sequencing. Right now, probably the biggest disadvantage, or maybe the biggest hurdle for this long read technology is, you know, in some cases they're costly. Certainly the case with PacBio, and they essentially just have a reputation of not having the same accuracy as Illumina. But that's something that is really no longer reality. So simply just have this as a transitional slide. You know, essentially, this is kind of where things start. We see these fish kills, or we see lesions on the outside of on the surface of fish, and the question is why, right? So if we focus specifically on using sequencing technologies, determining sequence types for the sequencing, is critical. If these are external lesions, do sample gills, sample skin. Something to consider is that that sample includes external environment. It's not just the fish. It's not just the target pathogen or target microbe. Certainly sampling individual internal organs or pooled homogeneous kind of cleans up the situation. Although all things considered, fish often have secondary infections, things to keep in mind. If possible, tissue culture, if we're looking for viruses, if a virus is indeed culturable, that's a great way to actually enrich a sample. Same for simple classical bacteriology type methods. We can sequence the colony right off the plate that we haven't identified using another method. That certainly simplifies the bioinformatic challenge that I'll get into a little bit. Sample preservation. This is critical, and it's really important to kind of have a plan in mind. I know a lot of this seems like common sense, but oftentimes samples are simply thrown in the freezer, or the downstream workflow may not be fully considered, especially in the case of an unplanned fish kill. So the question for sequencing is: do we focus on RNA? Do we focus on DNA? Next slide, I think I'll probably emphasize that RNA is probably the best place to start, depending on the question or depending on the level of unknown. I'm not advocating or I'm not endorsing products here, but we've had really good luck using DNA RNA shield in the field for stabilizing samples obviously with there. But that's certainly a product that's worth looking into. It's something that we had previously. So sample enrichment. As soon as we start doing this, we start adding bias, and but there are a number of reasons why we may want to do that. So probably the least biased approach here is using, if we're going to sequence RNA, is to deplete ribosomal RNA from the host. Basically, by doing this, we remove the host foreground, and this allows us to sequence RNA from anything that's essentially biologically transcribing its genome, which in active infection will be pathogens, as obviously as well as the host. Using poly-A capture methods, this is typically a standard method used for some of the aluminum methods, a lot of the kits, the 2C kits and whatnot, and if samples are actually sent to a core lab, typically if there's going to be a workflow used, this is typically the standard workflow used by a lot of contract labs. A drawback to doing this is not all pathogens, certainly some viruses, and basically don't polydenolate. So basically by incorporating this method, we start adding bias. This is just a simple workflow of RNA sequencing. I study RNA, and you can read this. There's really no reason for me to read this too much, but I do want to highlight 6A and 6B, and we'll talk about bioinformatics in a little bit. But basically, one thing that really enhances our ability to find new things is if we actually have a host, there's genome present or available for the host. We soak out those host transcripts first, and that simplifies the process. It's often not always the case, especially with non-model species. So sample enrichment for DNA. Yeah, so in the case of potentially asking questions specific to bacteria, we can essentially enrich based on methylated DNA. Bacteria don't methylate their DNA, so this is sort of the way of pulling out everything but bacterial DNA. We've been doing a lot of work with double-stranded circular viruses, and using rolling circle amplification is a great way to kind of bring those sequences into the foreground, enrich for those. And then, for questions specific to bacteria and fungi for that matter, basically doing some metagenomic library sequencing using shotgun methods, basically sequencing everything that's there, or actually using a targeted amplification of 16S RNA. Is a great way to enhance that foreground of the bacteria. Again, this a really simple workflow for those that aren't familiar with next generation sequencing. I think most of us probably are these days. Essentially, we kind of start with big pieces, break them up, and then try to find thematically put them back together. So really straightforward diagram here. So, really these days, sequencing is the easy part. While laboratories have MySeq or some other type of next-gen sequencers in-house, those that don't, outsourcing is a reality. There's many for many contract labs available to do the sequencing. It's really critical to actually consult and kind of work through the appropriate workflow that kind of based on some of those other slides that I mentioned. There is, no great one size fits all. Computing needs are significant for the some of these large data sets, terabytes worth of data. Is that something that my laptop would certainly handle? Many of us are not computer scientists and or programmers, really prefer to use Windows or graphic user interfaces. There are some bioinformatic programs that actually use a GUI, which simplifies things, but these programs are often limited. So for the most part, most bioinformaticists use Linux. Just some simple advice for for younger scientists that may be interested in doing this. This is the time to learn command line Linux programs and whatnot. If this is a direction we think we can to. So again, not endorsing any software. I did just want to kind of give some examples of some programs that are readily available. On the left-hand side, there are a number of programs that are available with a license and you're fairly costly with annual fees. I did just this week ran into a program called GAAP, which is actually free. I haven't tried it, but it may be worth looking into. And then there's a trove of open source bioinformatics software that's freely available. But again, it's primarily command-based. So sequence processing. In the case of next generation sequencing, when we receive gigabytes worth of data, it's critical that we're going to handle the processing on our own to try to clean things up and simplify them as much as possible. So as I mentioned previously, it's soaking out those sequences if there's genome or transcriptome available. It really simplifies our focus to working on non-host transcripts or non-host reads. Once we do the bioinformatic analyses, generate contigs. I'm not going to get into the details because it's kind of beyond this presentation, but it's basically we need to identify things by querying against a varied database. That's something that often we generate in-house. The novel pathogens, I primarily focus on viruses. It's critically used plastic searches, just protein homology is typically much better than sequence homology. And then there are some other tools like HH, right, to actually identify most related proteins, especially in the case of novel proteins. I simply included this slide to kind of emphasize that we're kind of at this interesting transition of identifying new microbes. This is obviously kind of specific viruses here. Many of these viruses are not similar to anything in databases, but databases are improving, and a number of these methods are allowing us to identify more viruses than really thought to look for. Some methods that I've been employing to kind of simplify this process, and this is one thing that's great about where we are with bioinformatics, and this is just a reflection of the need, is that there are some programs and workflows that have been developed. This one specifically called CNOT Table 3. I've used 2 in the past, but this is sort of the new version. Essentially, we can run our configs through this program, and it automatically annotates viruses based on homology of some highly conserved proteins. So, at least this process, you can become your own bioinformaticist or simply reach out to a collaborator. It really depends on your need. Becoming a bioinformaticist is not trivial. I think a number of us dabble in this, but when you really get serious about identifying things, I personally find it's easiest to reach out to some other collaborators. So in the case of bacterial pathogen discovery, and maybe I'm using discovery incorrectly here. This may not necessarily be new pathogens, but pathogens that we're aware of, but have emerged into a new host or simply into a new garden. Essentially, metagenomic analyses are probably one of the more reliable ways of identifying what bacterial pathogens may be present. There are a number of programs that are useful for this. Shine, Mother, Megan, and there are actually a number of professional services where you can upload your data and use some proprietary workflows as well. Simply included this slide. To put this on your radar. AI is sort of a big buzz right now, and AI does have a place in bioinformatics and pathogen discovery right now. So again, do we need to become a bioinformaticist, or will AI assist us in their future? So part of the reason for presenting is kind of pointing towards resources. This may be something some of you are interested in. I'm not sure how many of you have mined the SRA database looking for pathogens. A lot of folks, certainly during the pandemic, actually started mining in silico to find new interesting pathogens. So this is a tool that can certainly allow you to do that. Also, if you identify a new pathogen, if you run that sequence through Pebble Scout, you can potentially identify other samples that have been submitted in the SRA that actually have that pathogen present as well. So just to get a better handle on diversity and novelty. So these are just a few examples from work that I've been involved with. I kind of give you a little background on the piece here. This was essentially a sample from a Siamese fighting fish data. The investigative research community specifically had questions about mycobacteria. When we did the sequencing and bioinformatics, we ran this through Megan. And there are a number of pathogens beyond myco that pop out. And interestingly, if you look, there's actually host sequencing here of some from beta and others from trout. In this particular case, using this workflow is actually really useful as it essentially identified a contaminated sample. Most likely the sample was contaminated probably in the core lab, simply because both of these species didn't exist in the local lab and the samples were handled. Recently, I was involved in some work with Jan Levy. There was the chronic die-offs of menhaden on the Atlantic coast with a spinning disease. Historically, there had been some work done back in the 80s that had identified an ICAM-like virus. So we actually kind of started with specific primers for IHN and didn't identify virus. Tissue culture came up negative. These the fish, certainly had secondary infections, but we were kind of focused on this virus question. We stepped back a little, be a little more holistic with our approach, and using 16S metagenomic sequencing, basically identified highly enriched Vibrio angularum, particularly in the brain. So with some sequencing, we actually had pure colonies of Vibrio. After we were fairly sure that this just wasn't incidental co-infection, and sent samples off for sequencing a number of different samples, simply to get a handle on diversity, for which there really wasn't much of rare diversity at all. This experience was a game changer for me. We used a company that had a fairly inexpensive cost for sampling bacterial genomes. The cost was about $125. So we were able to, in this particular case, because we had cultures of the bacteria, we were able to grow up the bacteria in suspension and ship them off for sequencing. So that $125 includes DNA extraction, the sequencing, and bioinformatic assembly. And essentially the turnaround is about 3 days, which in many ways makes this appropriate for diagnostic labs and cases. So just something to keep in mind. We've noticed that same lab now actually offers a version of using oxygen core sequencing plus Illumina sequencing on the back end for not that much more. I'm not sure how important the Illumina sequencing is on the back end now that they're using the R10 sequencing or chemistry. But it still seems to be a request from the journals anyway. I've recently been involved with some work with large and smallmouth bass, which is called blotchy bass syndrome. We've identified a handful of adenovirus. Adenoviruses in fish, as well as a number of other viruses. Simply by using these next gen approaches and the C-note taker workflow. If you look on the left, there's sort of a simple screen capture of some of the viruses that we're pulling out of these fish. In many cases, we're getting near complete genomes of some of these viruses. And again, something that I want to point out is in the particular case of the adenoviruses, the replicase protein is similar to papillomaviruses, and that was our only hit. We were blasting the entire genome. There were essentially no significant blast hits for most of the other warps. So again, this is the case where we kind of are working in that dark matter sphere, when it comes to identifying these novel viruses. And not unheard of, especially these days with these de novo methods to actually identify viruses which family names are generally been described here. But a lot of these automated tools are not perfect. That's automation is great until it isn't. So there are a number of cases where they're actually making maybe hits from these tools that are not appropriate. In this case, there was a tail protein phage that actually, when we blasted it to NCBI, it actually turned out to be a host protein. This is just a quick list of some viruses that we've discovered using these approaches, essentially in the past decade. I don't really want to spend much time on this, but essentially, we've been finding more viruses that can actually really research. Recently, we did some work with I mentioned this that slide with the genomic analysis samples from a beta. There's the primary questions. Primary goal is to actually identify a mycobacterium, kind of in the process of screening contigs from the next gen sequences to identify a novel papillomavirus. We don't know if it's associated with disease or not. That's something that, unfortunately, a lot of these methods kind of direct us down that path of identifying novel things, fish that are sick, and simply because we identify something new that's a micro, something that is the disease. A number of years ago, we were involved with the project with the U.S. Fish and Wildlife Service. There's a fish called fountain darter, which is endangered. There's an active restoration program. And in this case, the Fish and Wildlife Service was routinely isolating a filterable agent that actually did produce CPE in cell culture. So this is a case where using cell culture to enrich to do the virus identification really became useful. So we essentially used RNA sequence for the case. Because of the cytopathic effect, we already did have a general, we suspected an virus. And so, and this is the case again, kind of going back to the enrichment or library prep steps. If we had used a poly-A enrichment method, we probably wouldn't have picked up viral RNA. I've talked to a few other investigators that have used that same pipeline and have not been able to sequence viruses anyway. So again, the less it's important to be cognizant of your library prep. So in this case, we actually did perform challenge experiments. This was an virus, and like most, actually to disease, or certainly not mortality. But these kind of steps, in this particular case too, because we actually had isolate hand opposed to just sequence, we could actually kind of follow up and kind of ask the necessary questions of the specific to potential pathogens. But these are steps that are not always possible. But simply using sequencing methods to identify new things is something we need to be careful of. We're going to ascribe disease down the road. So here's an example of a paracompativirus that we've identified with suckers. This was essentially pulled out of the RNA-seq library. We were trying to identify post-transcriptomic responses in white suckers inhabiting areas of concern in the Great Lakes region. You know, again, we found a new novel virus. It's kind of exciting. This is actually the first hepatitis B-like virus identified on avian or mammalian hosts. But simply because it's related to hepatitis B, it does not necessarily mean that it's a disease agent similar to hepatitis B. In the absence of actually having isolate or really humans to challenge experiments, you know, we kind of opted to screen lots of fish to get a better handle on prevalence. So we just, you know, essentially developed diagnostic markers, qPCR, and then again, in the absence of the option to do challenge experiments, we started doing transcriptomics on the host liver and other tissues, kind of get a handle on whether or not there is a host response indicative of disease. There's some of that work is still in progress, but you know, again, there's sort of this trap of identifying things and then trying to ascribe them to, you know, some type of disease state. Emerging adenoviruses. This is something I've been working on for the past 5 years. We've identified adenoviruses associated with these really interesting clinical signs of hyperpigmentation on black basses, smallmouth bass, largemouth bass. We would not have been able to identify these viruses without using these next-gen de novo sequencing approaches. So, you know, again, like the application of these tools is sometimes necessary, particularly in the case of viruses that may not grow in tissue culture or propagate in tissue culture. I believe we now have evidence that we can culture some of these viruses, but they don't appear to be cytopathic. So again, in the absence of a cytopathic effect, it looks like there's nothing there, right? So it kind of gets to that loss will to live diagnosis. Although I guess I should mention here, in the case of these adenoviruses, it's not clear if there's a real negative health impact on fish. So, you know, this is kind of where I wanted to land with this talk, is sort of, you know, kind of walking through workflows. You know, if you don't know why fish are sick, you know, using RNA-seq with a post-ribosomal RNA completion preparation is probably the most unbiased, closest approach that can be taken. You know, sample collection obviously depends on these presentations, something external, internal. You know, an RNA beading shield sort of been a recent go-to that I've used with high success. RNA-seq using the Illumina platform is sort of, you know, the standard, but you know, Oxford Nanopore these days seems to be probably the most cost-effective approach for long read sequencing. You don't have access to these sequencers, or have a colleague that has them. You know, there are a number of contract labs available that they'll do the sequencing. We've been using GenoHub, which is essentially sort of a broker for next generation sequencing services. Yeah, it's actually a fairly simple interface to use. Basically type in a description of what needs to be done, and then GenoHub essentially identifies labs that can do it and provide quotes. You know, so bioinformatics, So I didn't really lean into some of the complexities of bioinformatic analyses as much as I did to. But again, I think there's a critical reality check that we need to have when it comes to whether or not we want to be a bioinformaticist or something seek out an expert. I personally use CLC Genomics Workbench and Genius to kind of use that graphic user interface environment for a lot of the work that I've been doing. I have learned enough command line and dangerous and emphasize the complicated analyses that better collaborate. So just a few final thoughts here. I've kind of mumbled through some of this along the way here, but in the case of the de novo sequencing that we are now able to do, we're starting to identify a number of novel viruses that we really don't know what to do with them other than catalog them. Something that I think is really critical. Simply inventorying these and then making them publicly available so others can potentially make specific tools to screen for them in case of health effects. Depending on sample type and sequencing method, many non-host sequences will be identified. That's pretty obvious. But simply identifying these sequences doesn't necessarily, well, it certainly doesn't establish cause, right? So novel microbe is not equal novel pathogen. That's something that I think we need to overemphasize as much as possible. Simply because it's really easy to get excited about finding things. So there is value in documenting all microbial species associated with aquatic organisms, regardless of health status. It's a personal feeling of mine. There are a number of cases where this is kind of also specific viruses, or at least in the case of viruses, they may not be primary pathogens, but we're seeing environmental changes these days where a lot of where some microbes that were once not an issue are becoming problems in different environments due to those changes. Copy number for the abundance of the pathogen sequence data set. In some cases can infer a relationship to disease. I didn't mention this in the case of the beta papillomavirus, but the papillomavirus sequences that we covered were almost 2% of all of the DNA in that fish. And in the case of some RNA viruses, we see transcript levels of about 0.1% or so. That's often indicative of an active infection. So where do we stand today? Through a lot of classical fish health work, a lot of the easy stuff is done. There are likely lots of viruses out there, that kind of live in that dark matter category where they're so different than anything in the database that you can't simply find them, even with some of the bioinformatic methods. We're probably also at a state bioinformatically where we're probably going to start needing to evaluate the complete microbe environment. Polymicrobial diseases are real, and some of these approaches here can probably help us address those disease conditions a little bit more thoroughly than it's possible. Next generation tools are necessary to better characterize microbial pathology. It's being done commonly, be external to deficiencies, deficiencies. Certainly in the case of some bivalves, muscle die-offs and whatnot. These next gen approaches are critical for addressing these questions. These methods are rapidly becoming standard tools for biosurveillance of known and exotic emerging pathogens. And clearly, the discovery tool. One of the nice things about the next gen approach actually having that high fidelity sequencing is that unlike PCR, just with this one step, we have sequence level resolution. And that's really where I wanted to finish up. And something I guess I wanted to mention here too is if you employ these methods, we collect lots of contigs, we identify a lot of new viruses. Don't keep your pathogens a secret, don't keep your microbes a secret. There are options of publishing genomes, and certainly uploading them to public databases is critical to kind of help the collective community anyway. There's so much out there that we really can't just hold on to things. All genomes are no longer a publication anymore. It's something that we keep in mind. But I don't know. At that, I think I'll open this up to Nick and others.
Excellent overview. Thank you for the presentation. Folks, if you've got questions, feel free to drop them into the chat and I can read them, or you can unmute yourself, raise your hand and ask Luke directly. While we're waiting for those to come in, I'll ask you a question, Luke. Do you know if there are complementary efforts on the cell culture side to develop cell lines for these novel viruses that you may find through the NGS process?
A number of labs, I think, periodically try to develop new cell lines. I mean, that's certainly, the lack of appropriate cell lines is probably one significant reason that we don't identify more viruses via cell culture than we do. To be honest, I'm not clear how big of how much effort is being spent developing new cell lines, particularly because some of these other methods are now available too.
Mm-hmm. All right, Ademic, do you have a question?
Hi, Luke. Thanks for a great presentation. I have a question about how to approach this kind of data where you are isolating more and more viruses, and it seems that the situation is coming to mimic the bacteria situation. You are isolating bacteria and you don't know what you will always find some in every case scenario. And right now with the new methods, it seems that with viruses, we will have the same situation. From the data which you are generating with RNA-seq, you already mentioned that you are looking also on the responses of the host. Is it not better to sample a lesion and a site outside the lesion and compare what kind of immune responses you have in the site of lesion, and then somehow direct you if this is an antiviral response or not antiviral response?
Sure. Yeah, so I'm glad you pointed that out, and that's it's a great observation. And that is our reality, right? So and I probably should have emphasized that on the sample collection slide. I'll just give an example with this situation with the blotchy DAS syndrome we've been working with. So specifically to the approach that we're taking is we sample the lesion and we sample paired normal or clinically normal skin to the case. And we compare essentially the microbiome as well as the host response in those samples to kind of better address that question. But you're right. I mean, these data sets unfortunately become unwieldy and difficult to interpret. So, yeah.
Hey, Luke, that's actually a great segue into kind of my question. Unfortunately, Stephen Reichley's not here to corroborate this, but you know, the advancement of these technologies is insane, right? So when Stephen started his PhD program, it took him a year to do the first bacterial genome with primer walking and based on Illumina. And by the end of his PhD program, he was sequencing, you know, entire bacterial genomes in a couple of weeks. Where do you think we're going with this? What are going to be the limitations? I know we talk about the company that you and I have discussed, and the turnaround is insane. What do you think the future of this kind of genomics and next-gen technology is, and what are the limitations, you think?
So that's a good question, and you know, it's just, I think about this every so often because you know, even earlier in this presentation, you know, I kind of mentioned, you know, perhaps we need to be bioinformaticists or consider that. You know, I think potentially a lot of students, you know, getting a handle on that quantitative aspect is probably almost becoming a bigger need than some of the lab-based work. Although, you know, clearly addressing the question of relevance, especially with you know, when we have these differences in communities that may actually be associated with some type of health outcome. Clearly, we need, you know, we need to be able to perform challenge experiments. But you know, in the case of how to handle these big data sets, I mean, I think it's going to change the nature of some of the professionals that are part of the field, because it's going to be more of a data analysis driven approach, I think, to answer some of these trickier questions and really bigger questions. You know, and again, I don't know where AI comes into this, because that seems to be moving a little faster than on the great floor. Does that kind of address?
Yeah, well, there's no right answer to it, Luke. I just wanted to get your opinion on it, and
yeah,
I really thank you for helping us out here today.
Sure.
Say, Luke, go ahead, Jesse.
Thank you. Hi, Luke. This is Jessie from University of Idaho. Thanks for your presentation. I have a couple of questions. The first one I saw, like the Emily also asked in the chat in the chat board, and then so yeah, I have the same question, like the target samples, because you isolated so many different virus from different fish species. And also I assume like different virus, they have the different target tissues. So how you make a decision to take what kind of the tissue from the samples. And another one, like I want to ask, how common you find the co-infection of the fish? Because I think you mentioned like the bacterial and viral co-infection in some of your case. And then, so how can you figure out which pathogen is the primary or the first one, and then which one is the second, the following pathogen, and how you make sure like the virus or the bacteria is the major to cause the problem, but not like the another.
Right. Yeah. So I mean, that's a critical question, right? And I don't have a good answer to that, right? So in the absence, at least, how do you in the case of a co-infection?
Josh picked up attention 2172. Josh 2172.
So in the case of a co-infection, how do you ascribe which is the driver, or is one microbe more important than the other? Is it generally a polymicrobial disease? Essentially, I think it's a case where we build to be on classical challenge methods. Also, or if we sample enough and kind of see those relationships associated with that particular disease enough. I think by association, we can kind of get there, but it's not a robust. Yeah, I think Hill's postulates can be used in that particular case, but again, it's more of a weight of evidence approach than that experimental approach. As far as what tissues do you sample? I mean, typically, and I'll be honest, so a lot of these viruses that I had in that list came from fish that were, clinically normal. In many cases, we were sampling these fish for other reasons, and sort of, incidentally or opportunistically identified these viruses. So again, like it's not clear if they truly are pathogens, or if they're just a normal part of the vibrome of these hosts. That's something that, again, sort of there's probably going to be a paradigm shift of sorts, but probably not too far in our future. It hasn't been that long since all bacteria is bad, right? And our microbiome, or now the microbiome research is kind of advanced. You have a better understanding on how important bacterial populations are in specific ratios of different representatives of those groups. We're probably not too far off from having a handle on what may be a healthy or a normal virus. Not all viruses are going to be bad. In plant science, there's evidence of beneficial viruses. So I think we're just starting to peek at this other world that maybe we had a suspicion was there. We didn't really know.
Yes, thank you. And so another one, sorry, I have another. Did you try like an electric microscope and then to check the character of those viruses?
So in some cases, yes. So in the case of that white sucker adenovirus, we actually do have electron microscopy on plasma samples, and there was a significant amount of viral particles in those fish. We recently identified another hepatovirus fish called alewife, and virus is certainly present in a number of different fish. So it's not that's simply incidental finding, but when we've done EM on those fish, we haven't found particles. So but having that EM evidence, basically is evidence of replication of the proteins, right? We're not just looking at a genome. That's something I didn't mention either, but we've found evidence of integrating viruses into genomes, including herpes viruses, and in some cases, I think evidence based on sequencing is that those integration events probably happened millions of years ago even. So that is something to be cognizant of too, is that simply finding sequence that has homology to some type of virus doesn't necessarily mean that virus is present. It just means that there could have been an integration event, but we're simply looking at a host sequence, sort of those fossil sequences.
Great, thanks a lot, Luke. We're out of time and there's a few questions in the chat. So I encourage you to reach out to Luke directly. His email is there on the screen. I'm sure he'd be happy to follow up with you. Join me in a virtual round of applause for Luke, though. That was a great presentation. Learned a lot, very interesting.